Okay, so the next part of the chapter is part 2, 7.2. We're going to be talking about protein synthesis. So how are proteins made within our bodies? So first off, proteins, we know, are big, large macromolecules, and they're made up of these repeating subunits that we call amino acids, right? We learned that in the last video. There are 20 amino acids. Um, 9 of them are essential, which means we have to obtain them from our diet, and the other 11 are non-essential, which means that we can either obtain them from our diet or our body can make them. So proteins are going to be long chains of these things that we call amino acids. Okay? Amino acids get joined together to form these long chains through a process called dehydration synthesis. In that process, something called a peptide bond is formed, and also water is removed. So let's take a look at this figure. So here is amino acid number 1. Doesn't matter its name, right? Just amino acid number 1. And here is amino acid number 2. And so we can see that we have got the amino group, central carbon, bonded to a hydrogen. Here's that methyl group. And then bonded to an acid group. That's the first one. And the same thing with amino acid number 2. Again, we have the amino group, the central carbon here, that's bonded to a larger group, not just a methyl, but this is a sulfur-containing group. We got sulfur right here. And this is also bonded to a carboxyl group. Okay, so you got amino acid number 1, amino acid number 2. The acid group of amino acid number 1 is going to react with the amino group of amino acid number 2. And so what's going to happen here is water, so H2O, so an OH from this carboxyl group and a hydrogen from this amino group are going to be removed. So water is removed, okay, H2O. And so when these get removed, what happens is this carbon from amino acid number 1 is now going to covalently bond with this nitrogen from amino acid number 2. And so here we go. This was amino acid 1, and now it's bonded to amino acid number 2. So again, the carbon of the carboxyl group from amino acid number 1 is going to bond with the nitrogen from amino acid number 2. Okay, and this bond right here, it's a covalent bond. So a covalent bond is going to be a bond in which electrons are shared between these two atoms, and we give it a special name in proteins. We call it a peptide bond. So that process is called dehydration synthesis. Water is removed and a larger molecule is formed, okay, through this peptide bond. When we have 2 amino acids together, we call that a dipeptide. When we get more than 2, so 3, 4, 5, 20, 100, however big the protein is, we're going to call that a polypeptide. Okay, so single amino acids are just called amino acids. When you have 2 amino acids together, we call it a dipeptide. They'll be held together again via a peptide bond. And when you get more than 2, we're going to start to build a larger molecule, which we will call a polypeptide. Okay? Again, the process by which dipeptides and polypeptides are formed is called dehydration synthesis. Water is removed and a bond is formed. Okay? So that's how amino acids are put together to form a protein. But how is this dictated? And so this is a nice figure from your textbook that really sums up the process of protein synthesis. Okay? And so we're going to go from what's coded for in a gene, so the genetic message, the sequence of bases in a gene. And that's going to then be translated eventually into the sequence of amino acids in a protein. And that sequence of amino acids in the protein, again, is dictated by the gene. And that's going to be really important. That sequence of amino acids is going to tell us how the protein will eventually fold. And how it folds dictates its function. So its final 3-dimensional shape is going to tell us what that protein does. Okay? Now, in my general biology course, I spend a few weeks teaching this. And so we're going to sum it up really quickly with just a few slides. And so I'm going to warn you from now that this is not going to do it justice. But this is not general biology, and we're not diving too deep into this. And so I'm going to give you a basic overview. Okay, So in all of your cells, right, every single one of the cells in your body, this process takes place, okay? So in the top of this figure, where's my pointer? Where are you? Here we go. The top of this figure, this shows a cell. And again, there's information about what cells are in the back of your textbook if you need a reminder. This is the cell membrane. It's the outermost barrier of this particular cell. We then have cytoplasm, or cytosol as it's labeled on here. This is the fluid portion in between the cell membrane and the nucleus. And in here is the nucleus. And so inside of our nucleus, we have genetic material. We have chromosomes, which are made up of DNA. Okay? So what happens during protein synthesis? We're going to go from DNA to a molecule called RNA. And then that RNA is going to be read. And according to that message, amino acids are going to be put together via dehydration, synthesis, and peptide bond formation. And you eventually get a polypeptide. You go from a single amino acid to a dipeptide, to a tripeptide, to a polypeptide. Okay? So how does this happen? So number one, we're starting here in the nucleus. A portion of the DNA will be unwound and unzipped. Okay, so here is the DNA. This is the double helix. And the two strands of DNA will be unwound and unzipped so that they are single-stranded. Okay, and your cellular machinery that's involved in this process called transcription is going to bind to the DNA, the gene, and it's going to transcribe, copy the message into this molecule here, which is called primary RNA. It's a transcript. Okay, so that's the first step in protein synthesis, this thing called transcription, where we go from DNA to RNA. Okay, this primary RNA transcript will then get modified into the final product, which is called mRNA. That M stands for messenger. Okay, so this is, a mobile copy of a gene. The DNA can't leave the nucleus, and so that's why we need this mobile copy. It needs to go to the cytoplasm because that's where protein synthesis takes place, out in the cytoplasm or cytosol. Okay. DNA never leaves the nucleus, usually. In a dividing cell, it will, but we're not talking about dividing cells. So the DNA is going to stay housed in the nucleus, but the RNA, mRNA, can go out to the cytoplasm. Now, there are two other types of RNA made really the same fashion. There's rRNA, which stands for ribosomal RNA, and then there's tRNA, which stands for transfer RNA. And both of those types of RNA are also involved in this process, but the mRNA is the one that gets read. Okay, so what happens next? So we have our mRNA copy here, our transcript. It was made during the process of transcription. It's going to exit the nucleus through a structure called the nuclear pore. Okay, when it exits the nucleus, it ends up out here in the cytoplasm or the cytosol. And out here in the cytoplasm, the mRNA is going to bind to something called the ribosome. This is number 3. And the ribosome is what reads the message. And so that's blown up down here in this second part. So the ribosome is this brown globular thing. There's actually a large subunit and then a small subunit. This green thing right here says UGU, AUG, AGA, AGC. This is the mRNA transcript. So the ribosome is going to travel along this. It's going to actually read it 3 letters at a time, 3 bases at a time. And according to this message here in the RNA, where's my pointer? The ribosome is going to bring in the right amino acid. Okay, so the ribosome is going to see AUG, and it's going to bring the tRNA in that's complementary to this, which is carrying the first amino acid, which is always methionine. Okay, and I'm not going to get too deep into complementary base pairing. Just FYI, U always is going to code for A codes for U, G codes for C, and C codes for G. Okay, that's how the code works. Again, if you take general biology with me, we'll get into this very deep. The ribosome will then move down here and read the next 3 bases. So the genetic code is in triplet. It's always read 3 bases at a time. And so here we've got AGA, and that codes for the amino acid arginine. And so this molecule right here, which is called tRNA, is going to bring the right amino acid in. Okay, and what's going to happen next is these 2 amino acids, through dehydration synthesis, will get covalently bonded to each other. Okay, and they'll stay attached to this tRNA molecule. That means that this tRNA molecule is now done, so that just leaves. And the ribosome moves down the message and reads the next 3 bases and brings in the next amino acid. So this codes for serine. And so on and so forth. And the process continues until you eventually have the message completely read with a bunch of amino acids all covalently bonded to each other, again, through dehydration synthesis. Okay? This will then be released, okay, at the end of this. Once the ribosome gets to the end of the message, there will be a signal, a stop signal, a termination signal, which is going to code for, okay, let's cleave, let's get rid of the protein. We're done making it, so we can release that. And then the ribosome just kind of falls apart and releases the message. What happens next is this protein will then, this polypeptide, I should say, will then go ahead and fold up. And we're going to talk about folding on the next slide. So again, a very fast run through. So protein folding. This is an important part. Form equals function. And so the final 3-dimensional shape of a polypeptide is really important so that that polypeptide can do its actual job. Okay. If we think about enzymes work, lock and key. So the substrate, whatever that enzyme is going to act upon, has to be able to fit perfectly with that enzyme. Okay. The same thing holds true for transport proteins. Whatever they're transporting has to fit perfectly into that protein so that it can be carried. The same thing with immune proteins, so antibodies. Antibodies are very specific to what they're going to bind to. So proteins have to fold up properly in order to be able to do their job. Okay, there's 4 levels of protein folding. Okay, the first is the primary sequence of amino acids. And so this is just the amino acids are put together in a very specific order, again, via dehydration synthesis, as the ribosome is reading the mRNA. Okay, what happens next is called secondary. And so secondary folding, we have 2 structures that are formed, and only one of them is actually shown on here. This is called alpha helices. It's a spiraling. And there are also these things called beta pleated sheets, kind of a folding. And that secondary structure comes from interactions within the amino acids. And so there could be hydrogen bonding happening. There could be these things called disulfide bridges that are forming. There could be hydrophobic, hydrophilic interactions. So there's all types of interactions that are happening with the side chains and also with the backbone of the primary structure. Again, we're going to get this beginning of some folding. What eventually happens is the entire polypeptide, so the whole string of amino acids, will fold up into a tertiary structure. So this is the 3rd level. The tertiary structure, the polypeptide is completely folded into its final 3-dimensional shape. And sometimes this is the end. So there are some proteins that only exist in the tertiary form if they're composed of just one polypeptide. The quaternary, the 4th level, is when we have proteins that are made up of multiple polypeptides. Okay, and so in this example, this is hemoglobin. It's made up of 2 alpha chains and 2 beta chains. So it's actually 4 polypeptides that fold up into their tertiary form, and then they associate with each other. And it's that final quaternary shape that allows a protein like this, like hemoglobin, to do its job. And the job of hemoglobin, it's a transport protein. It's responsible for transporting oxygen in the blood. Okay, so form equals function. The protein has to fold properly in order to do its job. And a little bit more on shape. So again, form equals function. The 3-dimensional shape of a protein, either its tertiary or quaternary structure, tells us what that protein is going to do. Okay? How a protein folds into the tertiary and quaternary structure is determined by the sequence of amino acids in the protein, in the polypeptide. Again, three-dimensional shape, final conformation is going to be crucial to that protein functioning how it's supposed to. There are things that can damage protein shape and structure, things like heat, exposure to chemicals like acids, heavy metals, alcohol, and there's other substances. When a protein unfolds or loses its proper three-dimensional shape, its tertiary or quaternary shape, we call the protein denatured. Okay, so that's a term that you're going to see. Denature means to unfold, to not be in the proper three-dimensional shape. This can happen inside of our bodies. We could also think about proteins that are found in foods. Digestion actually starts before you even eat food if you cook it. So when you cook, let's say, for example, a steak, okay, meat, high in protein. When you cook that meat, the proteins inside of there are going to start to unfold. Okay, so again, that process of digestion starts, if you're cooking, before you even start to chew your food. And that, again, that's going to be this form of denaturation caused from heat. Okay. This just kind of wraps up protein synthesis. So again, we had it on that slide. There was a lot going on there. So I just wanted to repeat it one more time for you. So again, steps in protein synthesis, part of your DNA code, specifically the segment called the gene. There's many, many genes, right? Whatever protein is going to be the product, it's going to be that gene that gets transcribed into mRNA, and that happens in the nucleus. So the first step is transcription, where we go from DNA to RNA. So RNA is going to be a copy of the gene, a single-stranded copy of the gene, and it's mobile. It can leave the nucleus. Step 2, the mRNA leaves the nucleus. It travels to the cytoplasm. Step 3, the mRNA is going to associate with a ribosome, and the ribosome is going to be the organelle that's going to read the mRNA, read the code, and translate it. So there's that step, translation. It's going to translate it into a specific sequence of amino acids in a polypeptide chain. Okay? To do this, the ribosome has to bring the appropriate tRNA carrying the right amino acid in according to the message found in the mRNA. Okay, amino acids that get brought in get added to the existing amino acid chain. That happens via dehydration synthesis. Water is removed, peptide bonds are formed. Okay, when the synthesis of the polypeptide is complete, it gets released from the ribosome, and then that polypeptide will fold into its active three-dimensional form and maybe associate with other polypeptides. Okay, so again, that's the summary of protein synthesis. And that's it for part 2 of the chapter.